It's really exciting to be here. I was hoping to come along and listen to some exciting science, and the opportunity to chair was also a nice surprise. I work at the University of Leeds in the IPRD. There's some nice friendly faces here who either used to work here or do actually still work in the IPRD. I'm their chair of digital and materials chemistry, so this is really relevant to me. It gives me the opportunity to introduce 3 exciting talks. The first, I think, is at the back, which is quite brave because you've got to walk down those really steep steps after. But it's Professor Andreas Bender. He's from the Department of Statistics at Ludwig Maximilian University Munich. He works at the Chair of Statistical Learning and Data Science and as a senior consultant in the Statistical Consulting Unit. Just as a bit of background, he obtained his PhD in statistics before doing a postdoc at the Big Data Institute in the University of Oxford, working on spatial analysis in the context of infectious disease mapping, and then as an interim professor at the Institute of Statistics at the University of Ulm, which from personal experience is a beautiful place to be. Currently is the coordinator of the Machine Learning Consultancy Unit, leading the focus group on machine learning in survival analysis. And he's also co-founder of an open science initiative in statistics and the PhD program coordinator at the Munich Center for Machine Learning. So really excited to hear what Andreas has got to tell us. So here, Andreas.
Yeah, thanks for the invitation to speak here. So what I will discuss is, and so I'm actually a chemist by training. Statistics is difficult for me as well. I'm a chemist by training, so basically I care about the data. Which types of data do we have? What does it tell us? Because right now in drug discovery, so that's the field where I come from, so where we have compounds and biological effects, there's quite a lot of push from the machine learning and computer science side. And that's a good thing, right? I mean, because there are great new algorithms, that's a really good thing. But chemical and biological data is quite tricky. It's really not trivial to deal with that. Yeah? And so this talk will reflect on that a bit. What does our data actually mean? What do the labels in the drug discovery context or biological effect context, what does that actually mean? So it's not so much engineering, it's more drug discovery, chemical and biological data. What are actually the perils of that? So I will reflect on that a bit, step back a bit, yeah, and to really think about things. Yeah? It's easy to just implement and use tech push, yeah, to do cool new stuff, but you want to use that for something useful as well, right? To really answer questions. And so I'm going to reflect on that a little bit for the next few slides. So any statements I make, because I will talk about our companies as well, but it's not a company message. So any statements I make, they are in my capacity as an academic, yeah, to step back and reflect on that, to try to do good science. And there are 2 articles we published as well in Drug Discovery Today a couple of years ago. They are shown here. And especially if you come from a computer science or machine learning area, and you're curious about what does data in drug discovery actually look like? What are the difficulties with that? Why did machine learning not have the impact in the drug discovery field that it certainly had in image recognition, yeah, or generative AI when it comes to certain other areas, yeah, or speech recognition and so on. Yeah. Our data is different. Drug discovery data is fundamentally different. I will explain that on the next few slides as well. But if you want to read up and go through that in a more systematic way, read those articles. They were picked up by Derek Lowe, who's actually writing a blog in our area. And then as a result, I was invited to talk to Angela Merkel, our then Chancellor, and Bill Gates and so on, to try to explain them the perils of AI in drug discovery. I'm not sure they understood any of that, but I can recommend, because you're good scientists, right? Angela Merkel is a chemist as well, by the way, but you're good scientists, so read those articles if you're interested. Yeah, they try to explain in a general, understandable way what's difficult about the data we have. So that's what we'll go through. Current state of AI in drug discovery. How do you actually validate methods? It's not as trivial as it sounds, yeah, because not just about getting a higher number, problem solved, we are done. It's not as easy. I will talk about data. Which is, in my opinion, the Achilles heel of data and, of AI and drug discovery. Not so much the algorithms, not the hardware. Yeah, it's actually the data we have and how we can label them. I'll come back to that. I will give a few examples of models that I think do contribute to the drug discovery context, especially, and that's a very important term, in vivo relevance. Okay, you don't just want to have a machine learning model that gives you good numbers. You want to have something that translates into the clinic. You don't discover drugs in order to target a protein. You want something you put into a human or dog or other animal or in a plant and so on, and that has an in vivo effect, right? But that is not as easy. I will talk about that. We started a few companies as well, so that's kind of a summary message. Lessons learned from starting a few companies, 2 here in Cambridge, 1 in Berlin and London. And then there's a few slides on, well, psychology in the area, so what actually drives decisions. So it's not that many slides overall, so don't worry about that. So where are we right now, AI in drug discovery? So it's what I would characterize as a third wave of computers in drug discovery. There was one in the 80s. Here's a Fortune cover from 1981. The next industrial revolution, designing drugs by computer at Merck. So it started 40 years ago, 50 years ago. It was very graphical. Doc was published. It's a docking program in the first iteration. It's still around today. Silicon Graphics was started. Some of you of the older generation might remember those machines, Octane and so on. So the first machines that did proper visualization of protein structures and so on. Then there was a wave, so very graphics driven, very structural. Then there was a wave around 2000. That was much more data driven. There was the biotech boom, 1999-2000. The human genome sequence was finished, the draft version, I should say. That was very much more data-driven. Yeah? So in the 80s, graphics, visualization, around 2000, much more data-driven. And then there's, of course, today's wave, right? Here I compiled some recent headlines. I mean, they are from a few years ago, but honestly, they still sound the same, so I didn't need to update them. How AI is changing drug discovery and so on, right? So it sounds quite similar. To 1981, right? But certain things certainly did change, right? We can generate data on a much larger scale. We can do spatial transcriptomics, for example, embryo development. Yeah? You can track that. You can do single-cell sequencing, spatial transcriptomics, and so on, right? So we can generate data. We can annotate it better. So when I did my postdoc at Novartis, that was 2008, 2009 or so, they started to clean up their own databases. Yeah? Before, big pharma companies didn't know what they actually knew. Yeah? There were no databases with standardized identifiers and so on. So even a company didn't know what it knew. It's a bit better today. It's not perfect, but it's at least a bit better today, I think, right? So that changed certainly as well, right? We have powerful algorithms, we have GPUs, TPUs, and so on. But the thing that did not change, and I think that's the biggest problem, is still the question, which early stage assay tells me about in vivo safety and efficacy? Okay, in drug discovery, it's not about finding a ligand, yeah? It's about putting that ligand into an in vivo context, very complex system, right? You have cell level, you have then heterogeneous cellular systems, organ level, and then the organism level, right? And maybe that's where it's different from areas such as catalyst design or aspects of engineering, right? You have an in vivo system which you don't completely understand. Yeah, and where everyone is different, right? On the genetic level, you have an overexpression of CYP, a different isoform, and so on, right? And quite often we cannot characterize the system. Yeah? So basically in drug discovery, we're always poking in the dark a little bit because we cannot properly annotate and characterize our system. And it's very different from other areas, okay? It's different certainly from materials design, where often there are underlying physical principles, yeah, that govern what happens, yeah? Or catalyst design, but it also goes into that direction. In the biological context, we don't know our system, okay? We can empirically describe it, but we cannot completely characterize it. Yeah? Even if you do single-cell sequencing, right? You don't do it on a whole organism level. If you eat something else tomorrow, it changes gene expression half an hour later, right? You cannot completely characterize the system. It just doesn't work. Okay, so and that actually did not change. So what's the early stage? What's the early stage assay that I should do to tell me about in vivo safety and efficacy? Okay, that did not really change. That's a fundamental problem, okay? Because we're still trying to identify that. Yeah? In some sense, I don't want terabytes of information. I want 1 bit of information in the computational sense, 1 bit. Which compound should go into clinical trials? Yeah? Just put that to 1 if it's the right compound at a given dose and so on. Yeah? I don't want terabytes. That's not good for decision making. Yeah? Too much information is no information, right? I mean, that's another way to say that. Okay, so certain things changed. We can do much more. But what is actually the crucial variables in our system that we need for decision making? That did not change. That's the point here. If we think back to 2000, so this is the time when the human genome sequence, draft sequence was finished. And 2000, there was published Craig Venter's article and the public sequencing effort. And this is from a review that was published the next year. And these are the expectations. And I show that to illustrate how high those expectations can be, maybe comparable to today, right, to the AI area. The number of drug targets will increase by at least 1 order of magnitude. Target validation will become a high throughput process. There will be more drug targets, 3,000 or 10,000, wherever those numbers come from, compared with, in those times, 483. Didn't quite happen, though. So there was a more recent article by Tudor Oprea, John Overington, and so on. So they estimated the number of drug targets, not at 3,000 or 10,000, but 667. Okay, so it's easy if you're in the midst of something happening and everyone tells you that's a best thing since sliced bread, I think is the saying. Yeah, it's easy to overestimate then, right? But you need to reduce it to practice, right? So if you have data and algorithms, reduce it to practice. And this is what I'm always trying to do with the group, also with the companies we started. How do we go from technology and potential to applications, right? So what are the limitations of what we do and that we need to keep in mind? Yeah. I will come back to that topic a few times later as well. There's quite a lot of funding going into AI and drug discovery. That's most recent numbers I found up to 2021. Of course, more recently than funding had its problems, right? There was much less money going into biotech in the last 2 years or so. It's difficult right now. I think around the end of the year or so, there will be a really crunch time for many startups that are venture capital funded. But the point is, there was quite a lot of investment in the area in recent times. Precise number doesn't matter, but it's a lot of money, okay? So that's what you put in, right? Then the question is, what do you get out? And so this is the discovery pipeline of AI-based startups versus big pharma. So here, startups that are what they call in this article AI native. So it's 20 companies that say, we do AI first. Okay, and you see that the vast majority of projects is in discovery or preclinical, so test tube stage, if you wish, right? And you have very few projects in phase 1 and 2, and basically nothing, or there's no the first projects that move into phase 3 of those companies. There's one very small trial for niche indication, but it's currently not drugged by Recursion. But that's about it. So you have very few late-stage clinical trials. In big pharma, the distribution is quite different. So this in green again, the discovery stages in phase 1, 2, 3, right? And in the drug discovery sense, phase 2 is the first efficacy phase. Okay, so first phase is for safety. Then the second phase is efficacy small scale. Third phase is efficacy large scale, usually. I mean, you can have different readouts, but in principle, that's how it works. And basically, if you don't have anything that went into phase 3, right, that means that this efficacy proof point is actually outstanding, right? That hasn't been shown yet, right? And given we already had 10 years or so to apply our algorithms in this context, yeah, that is something that needs to happen sooner rather than later, in my opinion. So those plots come from this Nature Reviews Drug Discovery article. There was a welcome BCG report more recently as well, but the numbers are pretty much the same, right? So the in vivo proof points, they still need to come sooner rather than later. You might remember the headlines I was showing 2 slides ago or so. So that's a kind of fun fact update 5 years later, right? So 5 years ago, AI was already changing drug discovery. 2023, we are inside the nascent industry of AI-designed drugs. And this is what some of the people who started the big companies in our field said, and that's quite realistic, in my opinion. There's no shortcuts to drug discovery. These trials are still in their early days, and so on. Yeah? So things get difficult if you go in vivo, okay? And my point is, if you think about a goal you want to achieve, it's easy to have proxy goals, yeah? But if they don't translate to the in vivo effect, they're worth exactly nothing. Okay, so see what your proof point is, how you validate a method. Yeah, if your early stage proof point isn't predictive, it doesn't matter. Okay, and that's something to keep in mind. So what's the conclusion? First part, there's lots of activity in the early stages of pipelines of AI first companies, but they are, as has been shown in those articles, already explores targets, close analogs. There was a more recent article by Boston Consulting Group as well that claimed phase 1 in AI first companies can go up to 90% success rate. Yeah? But if you look into that in more detail, you see that it's actually targets that have been explored for decades before. And of course, if you do something again 20 years later, where you had 30 projects in the clinic already and you know why they failed, of course, then you can achieve a much better result. Okay, so that's trivial. Yeah. So there's actually not as much novelty as we would hope for. Yeah? The reason is probably data is a limiting factor, and you can use data in 2 ways, right? One is empirical modeling, and then basically your model is limited by the data. Or you have something that is based on an underlying model, which you parameterize, yeah? Smaller hypothesis space. There are many good reasons to have a model where you need to understand something, yeah? Where you parameterize. Then you can extrapolate better. Difficult in biology, okay? Difficult to impossible even in biology, okay? Because you don't have many principles you understand, so you cannot break things down to a simpler model that you parameterize, so you cannot extrapolate, okay? That's the logical chain here, basically. And always keep in mind, if you read something, right, if you read news, is input success or output? So this company received 100 million in funding, or you get 3 billion and so on, or is it output, right? In some sense, telling a nice story and getting money is nice, nothing wrong with that, right? But you need to do something with that, okay? And it's the output that counts then, of course, right? And so, as a good scientist again, so the first AI-designed drug will be celebrated by the media, but as a scientist, you compare to the null hypothesis, right? What's the null hypothesis? To bring a drug onto the market costs $1 billion or something like that, right? So if you had 16 billion or whatever going into funding, going into the area, you would expect 16 drugs or so to come to the market, right? So there's a certain bias in the public perception and how those stories get phrased, right? So you have to be careful, right? Always compare to the null hypothesis. What would we expect otherwise? And people have analyzed data for decades. Yeah? This article from 1981, for example, right? Patient stratification bioinformatics, that's been going on for really decades already, right? In my opinion, it's a good step forward. What happens right now? We have new algorithms. We're able to build models we couldn't build before. That's very good. But it's in some sense more incremental in the drug discovery area. Other areas, there was really, I think, a quantum leap in my perception. In drug discovery, very difficult because the translation to in vivo doesn't really work as well. Yeah, it's easy to think if people do things in a certain way right now, that's how you do things, right? No, it's not like that. If you think about drug discovery right now, it's seen as a process. Yeah? So you have a certain compound, you want to have certain properties, and if you can tick all the boxes, then you say the compound goes into the next phase. Okay? People have done things very differently in the past, and you can do things in any way you want in some sense. So this is an article about Paul Janssen. So he started Janssen Pharmaceuticals. He and his team discovered 80 drugs in 40 years. No one else was able to do that. Okay, 80 drugs in 40 years. Not even a whole company within 100 years usually is able to do that. Yeah, and so he has run drug discovery as a very human-centric, in a very human-centric way, and not as a process. Okay, so sometimes you see those transitions from one phase to the next and so on, but that's all so simplified it's not even true. In practice, if you have a compound, you need to go back and tweak things, and then you try a few other things. Yeah? There's no linear process. It doesn't exist. And I think to simply get another view on how you can do things as well, articles about Paul Janssen and his approach to drug discovery can be very well worth a read. So validation. So what is validation? It's a core question in science and for startups, right? And if you validate a method, you might think, okay, I label my data, training test set split and validation, and then I have higher numbers on the test set. My method is validated. Okay. You can certainly publish an article, but is your method validated in the practical sense? Completely different question, okay? So, and especially in the drug discovery context, it's actually very difficult to validate a method. So in theory, we establish a method, use a benchmark, and we know how well the method works. That works if you have enough data, the data samples all the space you're interested in, and you're actually able to label your data, okay? And none of that is true if you think about in vivo relevant drug discovery data. So why is that? But it is nearly true, I would say, when it comes to ImageNet competition or speech, for example, right? You have huge data sets. They span everything that humans have ever said or written on the internet and so on. So you're getting as close as you can get realistically. In the drug discovery area, it's not the case. So labels are either mostly in vitro relevant or conditional. They depend, for example, on dose and so on. Yeah, makes it very difficult to label your data. I will come back to that. You cannot say a compound is toxic or not, depends on dose. That's a simple example. Validation is costly, so you can do hardly any forward validation. I mean, if you have a phase 2 study, that probably costs you 20 million or so, so you have only a small number, depends on the indication, so you have only a small number of cases which you can validate. So you have little prospective data, and it's difficult to impossible to sample the distribution in chemistry space well, okay? Very different from biological, from protein sequence space. There we have mutations, right? We have evolution. So we can see how likely is it to have certain transitions from one amino acid to another. Then we have, in the old times, the Blossom matrix or something like that. Yeah? And then we basically can make sense of the underlying structure, of likelihoods, of biases, and so on in that space. Doesn't exist in small molecule space. None of that exists, okay? We can estimate small molecule space up to molecular weight of 500 is 10 to the 60 or so, yeah? But that's about it. It's a rough estimate. Could be 10 to the 40 or 10 to the 80, yeah? So we don't know even that. And we don't know how are compounds distributed and what actually matters. What of that actually matters? What are we actually talking about? Thermodynamically stable compounds, compounds we can synthesize in a certain way, and so on. So my point is, what we can do in biological space, namely to understand statistically the underlying structure of the data, or in the catalyst space, material space, to understand things from the physical side. Yeah? So we have essentially mathematical equations that describe the majority of what we want to understand. None of that is true in the biological area. Okay, that's the problem. There's an illustration of that. So validation in chemical space is tremendously difficult. Why is that? There's practical reasons, but also theoretical reasons. So if you train a model, you have a training set, and every galaxy here is a set of molecules. Okay, so the stars are the molecules themselves. Every galaxy is then the chemical series or so. So you have a training set that covers a certain part of chemical space. You have a validation set. Then you have your external test set, which often is not really external. That's why it's overlapping here, right? It's drawn from the same underlying distribution, but let's call it external, right? Not true at all. Yeah, because then your next project is somewhere else in chemical space, okay? And what does performance on that part of chemical space tell you about those projects? Nothing, absolutely nothing. Yeah. So as opposed to sequence space, we cannot characterize chemical space, so we cannot quantify biases and significance. And the problem with the models is then that the data is insufficient to understand the mutual impact of the descriptors on the output variable we measure. So basically, if you have a chemical and you have a scaffold, if you go from methyl to ethyl in one case or another case, has a different impact. Yeah? That's a simple illustration of that. Yeah? It matters where you are in absolute space here. So it's a vector space. Many models, they have insufficient information to really understand if you go from here to here, yeah, what happens to the bioactivity, let's say, of your molecule. We don't understand that. And it depends on absolute distances, yeah, absolute locations and vectors, not only scalars. Yeah, it's a vector space basically. Okay, and there are those sayings here which I completely agree with. Every model is a local model. Okay, so maybe you can model something here, but you cannot extrapolate in most cases, and that also makes applicability domain concepts really local. Yeah. Sometimes there are attempts to estimate how well my model works in a different space. Well, it entirely depends if you jump here or here, okay? I mean, here there might be humans living, and here whatever, other things, yeah? So, and the model doesn't know that. The model doesn't understand that, okay? So basically, I think we should be realistic in our space. What are we actually able to model, okay? In chemical space, except in some areas where we know the underlying variables that matter. Log D, for example, is very well. I would even say we can model log D rather well. If you look into a big pharma company, you have a few 100,000 data points for log D, for example. Bayer published a paper on that, and that works well. Log D, we understand because there's physical properties that we can parameterize. Yeah? So we have a model that actually predicts and even extrapolates in that space because the descriptors capture the underlying trends in the data. It's usually not true in many other areas, such as bioactivity. There's also problems we caused ourselves. So this is, if you work in academia, this is what you're concerned with in many cases. So you have input data, you want to build a model, then you predict something, you have a confidence, and then you have your recall or precision or AOC or whatever you want. It gets higher, you publish your paper, and you think you get cited a lot and you achieve something. Not quite, because there's more to it, right? There's a project context, and there's the context of how does that actually get followed up in practice. So if you build a model, let's say, for cytotoxicity at a given threshold, yeah, 10 micromolar concentration or so, and your model is 99% correct, you might be very happy. But in practice, you work in a project context, and the compound might get dosed at a very different dose. So what does your model tell you? Nothing, right? Because it's not applicable. You have a different in vivo concentration, okay? Sometimes you have a project where you work in late-stage cancer, sometimes in lifestyle disease, okay? Which side effects are tolerated? Very different again. If you train the model, you don't know about that, okay? So you do this here, get a good model, okay? But your success criteria, your performance criteria are very abstract. They are not relevant for the really applied situation, okay? So there's a problem here. The other problem is, and I was working for AstraZeneca in silico safety for a while, and that's where I've realized the importance of this interface here as well. So you predict something, and what happens next? If that's all you have, the answer is nothing. Yeah? Imagine you spend 10 million on a project, and the computer says, oh, 83% likely that compound is toxic. What do you do? I mean, you scrap the whole thing because the computer says that. It doesn't happen. Okay, so you need to have a follow-up assay. So the question is, you predict something, and the next day, you need to run an assay that's cheap enough, fast enough, predictive enough, okay, to actually follow up on that. Okay, so you need to care about those interfaces. Project context, that needs to be consistent with the model you generate. And if you predict something, what next, right? So you need to have something actionable out of that. And that's very important to take into account, okay? It's not just about making a prediction. Yeah? It's embedding that into context to improve the process, if you want to see it as a process, okay? Here I used the word myself, yeah? So model, that's the process, yeah? Where you use the model in the process of making a decision, right? And for that, you need to look at those interfaces as well. Yeah, what to watch out for in validation? Many things can go wrong, right? There's quite a lot of proof by example around without baseline, irrelevant endpoints. In our area, there's lots of models for, I don't know, QED. So there's a quantitative estimate of drug likeness, yeah, that is based on historically approved drugs, but is completely irrelevant for future compounds that come from different chemical classes and so on. Or there's also some papers, generative models, that want to maximize log P and those things. Yeah, that doesn't really make sense, right? So relevant endpoints and optimizations about validation that matters needs to include the process, not just the model. It's tempting to ascribe success in what you do to the model, failure to other aspects, right? But of course, it's not the case. If you do drug discovery, you have a million small decisions, right? What do you know? How do you know what the contribution of the model was? Well, you don't know. Very difficult to do. Often there are small numbers and there are trivial successes, let's say biosteric substitutions. There's no negative control and so on. And this is the quantitative, or that's a list of things that illustrate why having generic performance measures is misleading at best, yeah, like an AUC of your model also, because that's virtually never what you need for decision making. Yeah? So here are some example settings that can happen in the drug discovery context, also in other contexts. If you come from a different area, usually there's an equivalent, just translate that mentally then. So sometimes I have a situation where I can select many compounds out of an even larger space, and I want to find active compounds, for example. And what matters there is more hit rate at the top. Yeah? So it's not the AUC across the whole curve, it's a hit rate at the top, let's say. Or I will come later to a pharmacokinetics model where we wanted to remove compounds with high clearance. Yeah? So it's one end of the spectrum that matters, to remove rubbish, for example. Right? Again, the AUC doesn't matter. Yeah? It's how good are you at removing the rubbish matters. Or you want to flag risk, for example. Right? So you have to see what is your practical use case, what is the performance measure that matters. That's what you need to take into account, and the interfaces, right? Otherwise, the model isn't worth as much. You cannot really translate it to practice. Yeah? And that's a problem otherwise. Yeah, let's talk about data as well. So what matters in the drug discovery context? The in vivo phase. So this is in this Drug Discovery Today article. So here we modeled the different phases of the drug discovery process, from hit discovery, preclinical. Here you have clinical phases, so you go into humans, right? Safety, efficacy. Smaller, larger. And what we modeled is being faster, being better, and being cheaper. Okay, and we mapped both shorter time needed, as well as savings on the same money axis, because you can quantify how long do you have patent life and so on. So if you make some assumptions, you can put it onto one axis. And what you see is, what matters by far the most is quality. So better compounds in the clinical phases, because those phases, they are long, they are costly, you have low success rates, okay? So the clinical phases matter. What you see in practice quite a lot right now is quite a lot of focus of AI in the early phases, finding ligands, okay? But we have 3,000 or so approved drugs. We have 50 million active ligands in PubChem and Campbell, yeah? So it's quite easy to see what's easier from that, right? So there's the tendency to look, that's the English saying, to look for the keys where the light is, yeah? Well, but the keys might be somewhere else, right? So we have to see what is actually the big question to answer. Well, this plot makes it abundantly clear, the clinical phases. Yeah? So the question is, what's actually the right patient that gets dosed in the right way with the right drug? Okay, so that means biomarkers, for example, patient stratification from the patient side, being able to extrapolate from animals to humans and so on for proper dosing. As well as from the compound side, even designing the screening library, even that factors in, right? So my point is, it's not the decisions you make here that matter. The decisions are often made before the whole thing starts. Yeah, what is the chemistry we actually want to use? Yeah, in our screening deck. Yeah? But all of that, cumulative success basically gets visible then in the clinical phases or not, right? So basically, the point is, in your particular area of research, which may or may not be drug discovery, ask yourself the question, what really matters to translate to practice, right? And quite a lot of effort in the drug discovery area is actually here. Well, actually, what matters is more those late clinical phases. There's two different ways to look at data. So one is what I sometimes call the flat Earth view, and the other one is more the round Earth view. So one is the case where we can label our data, or think we can at least. So that's more in vitro data, test tube data if you want. And the other one is the round Earth view. That's the in vivo data, which is much more complex and very conditional. And that's one of the problems we have. I've given the example before. toxicity depends on dose. I will give some more examples here that tell you how complex, how difficult that actually is in the drug discovery area. So this is how I thought about the world, my world, until 2010 or 2008 actually, until I started my postdoc at Novartis. Even if you're in academia, I can very much recommend do a postdoc, work for a few years in industry, yeah, just to get a practical view on things. When I was in Novartis, I'm a chemist originally, but I analyzed high content screening data, very biological data, omics data if you wish, but just imaging-based data. And I couldn't make sense of that. So it really, for 6 months, I was sitting there and I thought, hey, I'm gonna quit this postdoc, doesn't make sense, I don't understand the data. But what I realized is, well, you know what, Andreas, that's how biological data works, okay? It's very noisy, yeah, it's difficult to reproduce. That's exactly what you need to pick up. And this is what I always try to tell my students since then as well, yeah? If you think about the chemical area, biology, pharmacology, machine learning, make sure you cover all of those domains. Yeah? Don't just stay in one area, branch out to understand input and output and the tools you need. Yeah? So the chemistry, as well as the readout, whatever you're interested in, and the machine learning tools you need to change things in the input and then predict the output, okay? So make sure in your own research that that's the case as well. But this is how I thought about things. So you have a structure, you have activity on a protein or some mode of action, it doesn't have to be a protein. And here you have bioactivity data. Then your biology is organized in pathways or maybe networks, and it depends on the cell state and it depends on the cell type, but there's a structure to biology of some sort is my point. And then you have, that's linked to the phenotype, cell dies or so, and your phenotypic response data. And I thought, well, we can label those data and make useful predictions. And some of those allegedly useful predictions and labels are shown here. A compound is toxic. Compound B binds the target. Compound C treats the disease. None of that makes sense. Why? Whether a compound is toxic depends on dose, right? Whether a compound binds a target in the therapeutic sense depends on whether it reaches the target tissue, right? Target in the brain, well, it doesn't matter if in your test tube the compound is active, yeah? It doesn't reach the brain. Compound treats the disease, well, depends on disease endotype, right? Asthma, diabetes, any cancer virtually, yeah? It's a different endotype, it's a different class basically. If we don't label the class in the correct way, we cannot learn anything from the machine learning side. And quite often where we stand right now is actually on the very descriptive level, like 100 years ago. We don't understand disease pathways, okay? So we have those ICD-10 or other disease annotations that are very descriptive, often on the organ level, right? But we need to look into the biology. It's usually not done, even today. Yeah? So we cannot label our data in proper ways. And the other part is the data doesn't consider the conditionality in the data. And that's a real problem when it comes to bioactivity, drug discovery data in particular. There are many words on here. Just pick one or two examples that show how difficult it is to label data in the pharmacology context, for example. So if you have a compound that's active against the target or inactive, that's a far too superficial statement, right? Can be an agonist or an antagonist. It can be a partial agonist as well. It has a certain activity value. It has K on and K off rates. How fast does it bind? How fast does it diffuse again from the receptor, and so on. And those K on and K off rates, they're related to side effects in GPCR cases. Yeah? Often we don't know those numbers, so we cannot characterize our system properly. Okay, and that's true across biology, across pharmacology. Which pathways exist in a cell depends on the cell type, yeah? But there's a temporal element to that as well. It depends on the heterogeneous system you look at, and so on. Okay, so we don't know that either. So the overall point is here. Computational algorithms we have are very powerful. Hardware algorithms, no problem with that. But the question is, are our labels actually meaningful? A drug treats a disease. Well, it depends on endotype. Ligand is active against the target. Well, is an agonist, antagonist, partial agonist, whatever, and so on, right? So the problem is actually the data we have in our case is not so much computer science. It's are we able to label our data? And that's a huge problem. Illustration here about ketamine. It's just a more or less random compound. It's a very nice review article about ketamine in particular. So if you use a knowledge graph, for example, to repurpose drugs, and we tried that as well, then you need to label a compound with indications, let's say, and a dose and so on. So this is an example about ketamine, and how little we know and how much actually we learn over time, but that knowledge is still quite incomplete, and how conditional that is. So you can use it as an anesthetic, as a street drug, antidepressant, bronchodilator, right? Completely different effects. Maybe those two are related, but I mean the others are not. And that all depends on dose. So you can dose IV at 2 mg per kg or 0.5 milligram per kilogram, yeah? and then you achieve a different effect, right? It's the same compound, right? You just dose it differently. And you have different modes of action. Ketamine blocks the NMDA receptor, but there are also other compounds that act on the NMDA receptor, and they don't achieve the phenotypic effect, okay? They don't achieve that effect. Ketamine works as an antidepressant, okay, but other compounds that have the same effect, they don't work. So why is that? No one knows. Maybe the opioid system is implemented, and animals' metabolites are active, and so on. Okay, so the point is here, our knowledge is very limited. Okay, we learn quite a lot over time, and it's very conditional. Yeah, indication depends on dosage. Yeah, but it's not a huge difference. Fourfold, no, it's not so a huge difference, right? Another example: adverse reactions. Sometimes there are papers in machine learning journals, yeah, that try to say, does a drug cause an adverse reaction? Yes or no? 99% correct. And if you read such an article, it's completely useless in practice. Why is that? So in practice, it's not about yes or no. Okay, you have a dose that you give at a certain dose, a drug that you give at a certain dose, and then it depends on the genotype of the patient, who might have a certain disease endotype, who takes a certain dose of a drug, well, but maybe sometimes forgets it, right? And then you see an adverse drug reaction that uses a certain terminology that might not be unambiguous. And then you see an adverse effect in maybe 23% of cases at different severity, maybe only in males, maybe only if you administer with another CYP inhibitor or so, maybe only after 3 months of dosing. That's what real world data looks like, okay? So my point is, it's easy to trick yourself into believing, I build a machine learning model, my model is 99% correct, the nut is correct, yeah? But quite often that's not the case, okay? Because in reality, it's not a useful model, because you need to consider all of those aspects here, yeah? So does a certain drug cause an adverse event? I mean, it depends, right? It depends on all of those factors and even more. I mean, so even water can be toxic, okay? If you drink 3 liters of water in half an hour, you dilute electrolytes, right? So if you build a machine learning classifier for toxicity, would you put water into the toxic class? Well, probably not, right? But things depend, right? There's also some problems we caused ourselves. And that's a legacy of those high throughput experiments. In the biological area, I think in the physical area it behaves differently, but in the biological area, they've caused some trouble. There was the trend since the 80s or so to do high throughput screening in biological systems, right? So protein got isolated, you screen activity against the protein, and you hope if you go in vivo that the activity actually translates. Yeah? But the problem is quite often that's not the case. So, but the problem is you screen 1 million compounds, you have 10,000 actives. So what do you do now? Yeah? So you have another problem, and then you invent another set of simple assays that are not entirely predictive either, but they are cheap and fast, okay, to profile those compounds further. So we have now quite a lot of data that is actually not as predictive as we would like, okay, that is very high throughput, and that's the data that goes into models. And there's a problem with that, because they are not entirely predictive for the in vivo context. So you have an assay endpoint, what you measure, and then to an extent, you assume that that actually predicts something. So for example, you have a differential cytotoxicity assay in the glucose and galactose context, and then you use the ratio of those to anticipate mitochondrial safety. But there's only one mechanism, okay? It's very approximate. Huh? And so we have lots of data that is not as predictive for the in vivo situation as we would like. Yeah? And that's the data that goes into models right now. And basically, if you try to model biological data, always be aware, they measure only part of reality in a particular assay, in particular conditions. Yeah? And if you then use that and put it into a machine learning model, those limitations are quite often forgotten, okay? That was measured at a very high concentration or whatever, yeah? And that might not translate then to the real world situation. So that means there are actually multiple problems with that. So the interfaces we had before, right? The model is used in a context. So you need to consider input and follow-up. But there's also a problem even with the data that we have in the biological domain, I emphasize, in particular. That is very proxy, and is not actually the data we want in the first place to build our models. Yeah? So there are many problems with the current situation of using biological data in the drug discovery context. That's also one figure from this Drug Discovery Today article, trying to illustrate the differences between the image domain and drug discovery. So if you have images, so you have an object. So here, for example, a cat. And so you have a matrix, 24 bits or whatever at every pixel. So you can represent that. Here it's 1 bit, but the principle is the same. And then you can have a CNN or so that detects edges, for example. And at the end you have a bit, let's say that there's a cat or there's no cat, right? And it's very neatly integrated. So you have an object, you can represent it as a matrix, your CNN works on the matrix, and then you have a largely unconditional label. None of that is true in the chemical domain. That molecule is basically alive, right? You can have different conformations, might be reactive, you have different crystal forms, tautomers, and so on and so on. You don't know how to represent it, you don't know which machine learning method to use, lots of trial and error, and your label is conditional, right? And so that tells us that in the first case, when it comes to image recognition, there's actually very good integration of the object representation and the models, but it's far from true in other domains, right? So basically, there's no natural representation of the object in the first place and the application of the machine learning methods later. Yeah, so the areas behave very differently. And there's a danger, I think, with where lots of the attention goes, lots of the funding as well, when it comes to addressing some of the problems we have in the real world. There's quite a lot of focus over the last 5 years, or maybe even longer, on methods, machine learning methods and representations. You can do end-to-end learning, and that's all good, but that's actually based on something else. Yeah? And that is based, in this visualization here, on certain data that comes from a certain question or hypothesis. Okay, and I think that's a bit neglected, those parts down here in many areas. Yeah? And the question that people sometimes ask is, so what's the fanciest machine learning method we can use on that data set to just predict a label, okay? But it's not about label prediction, yeah? And it's not about, is that actually the right data for the right purpose, right? There's quite a lot of tech push in many cases, yeah? So data, let's say spatial transcriptomics becomes cheap, right? So everything gets sequenced, and then everyone says, okay, I have a new machine learning method, and there's this data set, no idea whether it's the right one, but my method is fancy and the data is fancy, so let's just do it, right? But the question is, actually those layers, they are based on each other. So the question is the question and the hypothesis I have, what is actually the data that I need to generate to answer that? How do I need to represent the data? And what is the method that only comes on top, okay? And you cannot save something up here that isn't in the data or that hasn't been generated in the right way in a relevant biological system, let's say, yeah? It doesn't matter what you then generate and which representation you use and which method, yeah? It was the wrong cell line or so, yeah? And then it just doesn't work, okay? So it's really important, in my opinion, to care more about reading this pyramid from the bottom, yeah? So to have a view of what is the hypothesis, what's the data I need, and then only the method and representation questions come afterwards. Explainable AI. So I went back to my old papers, I think from a PhD or so, where we also tried explainable AI. And that's an illustration of how it can go wrong when it comes to chemical data sets. So we tried to classify bitterness in compound structures. And this is and so we compiled, was with a company, 1,000 or whatever bitter compounds, and we tried to find chemical features characteristic for bitterness. Okay, and this is what we came up with. That's some example structures. If you're a chemist, you can see the problem already, I think. So here, that's some sample structures, and these are the features that come up that are associated with bitterness in the data set. Okay, well, but I mean, what is associated with bitterness here? Sugar rings. Yeah? So why is that? Sugars are sweet, right? I mean, they're not bitter. There's sugars that act on bitter receptors as well, by the way, but by and large, sugars are sweet and not bitter. And so why is that? Natural products are quite often glycosylated, so there are sugar rings attached, right? And natural products are quite often bitter, okay? Huge confounding factor or bias here in that data set, right? Well, and that's a problem with chemical data sets in particular. Quite often you have chemical series, okay, that you can easily make, for example, yeah, or purchase and so on. So there's lots of analogs in most data sets. Or you had a project where you explored a chemical series, yeah, and there's lots of biases in the data set, yeah, especially in the chemical domain. And so you have to be really careful. It's not because of the algorithm. The algorithm might be fine, but the algorithm assumes that you sample properly, yeah. Usually not the case, and so this is an example from our own work, yeah, where we were entirely naive and didn't consider that at all. Yeah? So you need to look at the data, especially if you deal with chemical data. Look at the data and understand the characteristics of it before you try any explainable AI or something. Yeah. A few examples where I think computational models can contribute for drug discovery. For example, we modeled pharmacokinetics in vivo data directly at AstraZeneca. So we modeled the in vivo endpoint. Okay, there's no extrapolation or so. We modeled the in vivo clearance and so on directly. Okay, that can be one way. So not sticking with some proxy is the point, but modeling what matters directly. Okay, so and I think that's a good approach. There's pull and push factors. So pull factors, automated DMTA cycles, for example. So there's the need to prioritize compounds from the pharmacokinetic side. They're very fast and so on as well. There's also push factors. data availability, so there's homogeneous in vivo data available from contract research organizations, ML advances and so on. And so overall, that was actually quite a beneficial project, I think, because there was a need, it's a suitable method, and there was some push because we had data, okay? If there's only push, that's not a good idea, okay? Always think about it. Is there pull and push? Yeah, and you need some pull, some need in the first place. And so that works in practice. Another area that I quite like is using gene expression data for compound selection. We did that to kill cancer cells, for example. It's a very simple concept. If you have a healthy state and a disease state, and in the disease state, let's say gene A goes up, gene B goes down, then you want to select a compound that induces the opposite gene expression profile to push the system back to healthy, basically. Yeah, it's not really mechanistic, that model at all. It's more pattern detection model, but in practice it works quite well. So we use that to differentiate stem cells, for example. You have a stem cell, you select a small molecule. After 3 weeks, you have a beating cardiomyocyte after the microscope. Really fascinating. I think that's the closest I came to feeling like God. Yeah, the computer and data. Yeah, you put a compound onto a cell, and then you see a beating cell. Yeah, you create life. Well, not quite, but it's really. I found that really fascinating. And that principle seems to work quite well. It's an area. It's actually a very interesting compound. It's an antihistamine, famotidine is called, that was shown in the clinic to regenerate heart cells after cardiac infarct. And people thought, oh, it's because of the histamine effect and so on. But according to what we found, it's actually because it induces stem cells in the heart to differentiate, okay? I think that's much more likely. We use it also in other areas right now. You might have heard of CAR T cell therapy. So what you do is basically you take a T cell, so immune cell, out of the body, you put in a receptor that then targets the cancer cell. So you use your own immune system basically to target cancer. The problem with that is it's a huge process. Yeah? It takes 100,000 euros or so to do that. You need to do that for every patient, yeah? Because you need to have T cells from that patient. But what you can also do, of course, is you can use small molecules to actually induce cancer cells in the body to express antigen receptors so existing T cells can target them. Okay, and that's the approach we try to pursue with this particular project here. So this is where we select small molecules to induce differentiation of cancer cells into dendritic cells that then activate their immune system, basically. Okay, so it's kind of the CAR T cell approach, just from the other side, if you wish. Yeah? There are many practical advantages. Small molecules, you have good pharmacokinetics, for example, easier, lower cost of goods, and so on. Yeah, few slides on companies and learning from that. Yeah, the real world is not only publication. Yeah, you realize that already. So it's if you have a publication, that's a good thing. But the question is always, how do you use that in practice? Yeah? So we started 3 companies: Helix, 2014 for drug repurposing in rare diseases here in Cambridge. Farm-Enable, 2016. So this was about accessing novel chemical and target space. So how can we make new molecules that target novel target classes? And more recently, Pangea Botanica. Well, originally was Terra Lumina, then Pangea Botanica is now renamed to Pangea Bio to do drug discovery from natural products and traditional medicines. And that's exactly to target this phase 2 problem. Yeah? The vast number of drugs actually fails in phase 2. It doesn't work in patients, basically. But we have lots of natural products with historical use annotations from plants. If you think about CNS active compounds, 84% actually have a natural product origin. Yeah? So it's good chemistry to modulate targets of interest, especially in the CNS area. Just to share a few learnings from starting those companies, because I think here in Cambridge in particular, some of you might have those interests. Yeah? Just to share a few learnings from that. So the location really matters. So this is from Helix. So one day, 2014 or so, Tim Gilliams, CEO of Helix now, came into my office and says he wants to start a company in data-driven drug repurposing. Result of the environment. Yeah, it really matters where you are. There's the original founding team was local. So Tim, David Brown, David Carvalho, and myself. There was Amadeus, so a venture capital firm basically next door. And then my students also working with or joining Helix afterwards, also AstraZeneca. Yeah? So location really matters, yeah? And those interactions that you can build locally. 2 learnings here that I want to bring across. It fell late in 2014 to start a company in the AI, or it was called big data in those times, yeah, in drug discovery area. Sounds ridiculous now, yeah, 2014. It sounds like 1250 or something like that, no? But I mean, it feels like that. If you do science, you're at the forefront of what happens, okay? And the world changes very, very slowly, okay? So basically, usually, things don't move as fast as you might fear, okay? So quite likely, if you're an expert in the field and you think it's a good time, it's not too late. That's very unlikely, okay? Certainly wasn't too late then. And building a company that is not only tech is safer. So Helix works with patient groups, for example. A venture company will always ask you, what's your moat? Yeah? So what prevents others from doing what you do? It's not going to be tech. I mean, if Google puts 100 people for 6 months on something, they will copy anything, right? So it's not tech. Yeah? You need a second leg, basically, yeah? Whatever that second leg is. And for Helix, is working with patient groups. In rare diseases, quite often they don't have someone to work with and so on. So it's quite a rewarding area, actually. Farm-enable. So again, there was this chemistry department factor. So we started with David Spring, Hannah and Natalia from the department. There was quite a lot of freedom from the Cambridge side. You can, on the site, start your company. It's great, actually, that is possible here, I have to say. No barriers whatsoever to set up the company. And there were local companies, Heptaris, or Zosa Heptaris now, or actually they renamed again, in order to validate that. Again, the environment really matters. The other learning is bootstrapping, so growing from revenue is tremendously difficult, and basically it doesn't work in biotech. We tried that for a year, very, very difficult, because you really need to invest to build tech, yeah? Otherwise you don't get anywhere. So I would not recommend it in our area. And the third part is Terra Lumina. That's now Pangea Bio, so the current company. So we try to start that in an incubator with a tech background, so not biotech, tech. And incubators can be useful, product market fit, something you iterate on 100 times, and it's actually helpful. It's a bit iterative, but it's still very helpful. So incubators can be very useful for that, especially if you do it the first time, I think. But eventually we decided not to proceed with that due to funding parameters. And there's always the question, big money, sounds good, but actually if you get money at a lower valuation, you don't want big money. You want money for 18 months or so, until the next inflection point, so where things really change. And in this particular case, it was too much money for the first inflection point and too little money for the second. So it was the wrong amount of money, basically. And it was tech money, so they didn't entirely understand biotech either. So what's the learning from that? Look for the right partner for what you want to do. So is it more platform and tech, or is it more bio and assets? And tech is entirely different from biotech. It's this West Coast versus East Coast difference in the US, basically, as well, right? And there are different viewpoints on validation. Is your platform validated? The biotech investor will never ask you that. He says, how good is your compound, okay? So basically see what you want to do and who are the people to talk to about that. Just 3 concluding slides or so. Then we are done psychology, so it's kind of a bit lighthearted, but actually quite serious at the same time. So AI is where it is, of course, also because of human psychology, right? There's quite a lot of fear of missing out. It's nearly entertaining to see. So when usually fear of missing out comes when your daughter is 14 or 16 and wants to go to the party, could have kissed Jimmy at this party or whatever. That's fear of missing out. And that also exists on the company level. Even CEOs of companies have that, right? Whatever, of a big pharma company, right? Our competitor actually works with that startup, Maybe we should do it as well. That really exists, It's embarrassing, but it's true, There's quite a lot of fear of missing out out there. Beliefs quite often drive decisions. Maybe they really have the secret sauce. I can tell you there's no secret sauce at all. The secret is doing it. And most people don't do it, whatever the idea is. Doing it is the secret sauce, which obviously is not so secret, right? So, and that gives rise to the ideal startup strategy equation. Hot air from startup, but lots of fear of missing out from big pharma, gives them the perception of the secret sauce, right? It's not really secret sauce, right? But if you basically claim something cool, don't tell anyone what you do, and then people from big pharma see that and they think, oh, maybe they really do, and that creates the perception of the secret sauce, right? Many things happen like that. So multiple levels, individual psychology as well as organizational psychology matter. There's also a few other drivers, of course. Everyone needs a winner, right? After investing 20 million, we need to show success. Imagine your boss gives you 20 million for AI, whatever, and a year later he asks you what worked. It's unlikely you will say, nothing. It's not, everyone needs a winner, right? So it's quite likely you will then claim wins, whatever they are. But the problem is, of course, selective reporting of successes leads to everyone declaring victory, but in reality, no one knows what's going on, right? So it's really difficult to advance the whole field if you cannot really compare methods. So to summarize, so we need to analyze our data, absolutely. AI is a valuable tool in the toolbox. In drug discovery, the real game changer will come when we understand biological data better, translation to the clinic. Methods need to translate into reality, so we need to go from model validation to process validation. And maybe the key take-home message, understand the data, understand what matters before you try to do cool stuff from the tech side. Because then what you can do is you can do cool stuff that really matters, also in practice, to really have impact in the field. I think that's the summary. Great. Thanks a lot for listening.